thank you for the organizers for inviting me to present. So today I will talk to you about what happened in 2023 in Uruguay. So there you go. So for the ones that are not familiar with wheat blast, here are some pictures to show you symptoms. And as Dave was mentioning, what difficult is this disease when it shows up in the field. Actually, one of the questions that still we need to answer is from where all this inoculum is coming. In 2 or 3 days, 200 hectare, 300 hectare fields are all blasted. So there is a lot of research to do on the pathogen cycle to understand what is happening. So as you can see, the pathogen infects on the rachis, on the spikes, and once he infects, it only moves to the top, okay? So it doesn't move to the bottom of the spike. That's a main difference with Fusarium head blight. And so this is, I took it yesterday from Javi's website. So this is what is showing for Magnaporthe oryzae Triticum distribution. I guess they need to update it after this talk. But so just a brief summary, it originates in Brazil in 1985, and then it spread to Bolivia, then Paraguay, then Argentina had 2 reports, one in 2007 and another one in 2012. There is also a report from US in 2011, but it was a single head. So I guess it's kind of unfair to, you know, paint all US in orange there, but we have one report. And then as you know, it moved to Bangladesh in 2016, and then Zambia in 2018. And those two, that intercontinental spread was caused by infected seeds. So this disease is a seed-borne pathogen, okay? And so of course, one of the questions was, well, look, Uruguay, no? I mean, for this one, there you go. So here is Uruguay, okay? So if you see that, you say, well, I mean, what is happening, right? Why we don't have wheat blast there? So a lot of hypotheses, right? Maybe the weather, we don't have the right weather conditions, maybe it's too cold, it's a tropical disease, right? It needs 28 degrees or more to develop. But then we have Fusarium, right? And they are pretty similar. And we also have a blast on rice, that is one of the main diseases there. So a lot of questions were we wanted to answer. And so in the meantime, 2 things were happening. So this paper came out from Diego Pequeño and CIMMYT researchers. So they combine wheat crop models with blast prediction models to predict future scenarios of blast occurrence. And so here you can see that Uruguay is actually getting a lot of red and orange. That means we were one of the places that we were in risk of getting wheat blast in the future. And meanwhile to this was happening. So in 2023, I hope you recognize this person here. This is Pawan Singh. So this is me. So AI is doing this, kind of. It's funny, but also a little bit scary, right? That it can, so it's the shirt and everything, right? So, okay, doesn't matter. So 2023, this was happening. Brazil and Paraguay were having a huge epidemics, like very, very bad epidemic. Like Dave was showing, fields of 400 hectares were all blasted at the same time. And so Pawan sent me an email, actually to all the South American colleagues. He was worried about wheat blast, right? And he said, well, Paula, what is happening in Uruguay? And we were actually right about to go to the north of the country to survey fields. Why to the north? Because in the north is where we have the climate conditions more similar to the ones that we have in Brazil. And so that's what we did. So in this figure, I want to show you first, in the gray dots is the winter crops distribution. So as you can see here, most of the wheat is actually grown in the southern part of the country, okay? Then I think it's sky blue, or I don't know what that color is. I mark all the states that we actually went to survey, okay? So these 4 states here. And so here is the border with Brazil, okay? So, and we don't have a geographical barriers, so it's just literally one street. So you cross and you are in Brazil, then on the other side you are in Uruguay, okay? And so we surveyed 11 fields, and from those fields we took 17 samples. So something that I want to share is that it was not easy to find it. All the fields were full of Fusarium. And that tells me a lot of things. So the first one is they coexist, but they coexist in the same spike? I don't know. I know they coexist in the field in different spikes, but I never saw them in the same spike. Then it also tells me that probably in those countries that Fusarium arrives first because it needs less temperature, maybe then blast doesn't have the space, you know, to come. But then, and also tells me about what is if we are doing good extension, because most probably in Uruguay, the farmers are treating as it was Fusarium head blight all the time, right? And they kind of know how to differentiate Fusarium head blight, but they probably were not aware of wheat blast, and they were spraying for Fusarium head blight, okay? So a lot of, because of this, we started to do a lot of extension with farmers and telling them, you know, please don't use your seed if you get wheat blast. Try to not bring your seed to the south, right? So we don't spread the disease southern, okay. So from those 19 spikes, we were able to, so we follow the Koch postulates, right? So identification, isolation, inoculation, and re-isolation, okay? So we got, in the microscope, we got 9 positive based on the spores characterization. And then we were able to get 4 monoconidial samples from those 9. And those 4 monoconidials were, we did DNA extraction, and we ran the characterization markers for MoT, the Triticum pathotype, and they were positive, okay? And then we moved to the phenotypic characterization, and you can see the typical symptoms. So here is a picture of the spores, and here is a picture of the seedling infection, and here the blasted spikes. So we did this under growing conditions using not biosecurity level, but biosecurity level 2, that is, we try to not put the spores outside. And so after this, we took those 4 isolates, and we took DNA and we sent them to K-State. So I did my PhD at K-State, so we have a really nice collaboration there. And so they did whole genome sequencing of 3 of the 4. So one of the isolates actually, it was not enough DNA quality to go through the pipeline. And so here you have a phylogenetic tree that we performed with the whole genome sequencing data using other 54 available data for other strains. And so I highlighted Uruguayan isolates in red and the B71 isolate, that is the one that caused the outbreak in Zambia and Bangladesh, in blue. So what we can see from here. So first, we confirm they are Triticum. they group with Triticum isolates and not with Lolium. So you can see in the right side of the image, the L on the beginning of the isolate name is identifying Lolium. Then we also see that Uruguayan isolates are different from the V71 isolate. That was the one that caused the outbreak in Bangladesh and Zambia. And we also see that among the Uruguayan isolates, 2 of them are clonal, so they share 99.87% of identity on the SNPs. But one of them is a little bit different. So 11.1 is actually 73% similarity with the other 2. And if you see in the map, 14 and 10 were collected almost next to each other, and 11.1 was a little bit further collected. And so this, something else that we see in this picture is that Uruguayan isolates group with isolates from 2012 and 2016 from Brazil. So these maybe tell us that we have had Piricularia or Magnaporthe for a few years already in the country. Maybe not. Maybe it's only appear in 2023. But it also tells us that about the aggressiveness and the difference in the isolates. So we don't have one clonal lineage. We have more than one lineage already in the country. And this was actually interesting because so if I want to show you here in this paper from La Torre from Sophie and Commons group that they actually instead of a phylogenetic tree they did a PCA. And if you try to put the Uruguayan isolates here, so 10 and 14 will cluster here, and then 11 will cluster here. So they are different. They are different to each other. And so we then wanted to check about mini chromosomes. So there are a few papers outside saying that all the variability in this pathogen comes from the mini chromosomes. And so when we compare the mini chromosomes from these Uruguayan isolates to the V71, that is the reference isolates, we found that for 14 and 10 there is no difference in the mini chromosomes. So here is all green and says that they are similar. But then when we compare 11 with V71, we saw a lot of differences in the mini chromosomes. Actually, some of the effectors are missing in 11.1. And that implies that this isolate has different virulence, mechanisms of virulence. So just to wrap up this, we found Magnaporthe Triticum in Uruguay during 2023 season. We actually think that this is a multiple introduction because we found similarity to 2012 and 2016 isolates. We have differentiated lineages already in the country. And all, of course, these show us what importance is to do surveillance, right? And how this disease will become a real challenge to wheat production. So we really hope that this disease is not adapting to cooler conditions, but already happened with striped rust, right? So we need to try to keep it in the tropics. So one of the, as I mentioned, one of the things we did is try to explain to farmers that the importance of not bringing the seed to the south, so we don't spread the disease in the country. So something that is still pending is, for example, as I mentioned, we did DNA extraction of 4 isolates. So the other one is 15.1. And these isolates actually in the greenhouse showed different patterns of aggressiveness. It was the most aggressive in the spike, but it was less aggressive in the leaves. And so, unfortunately, we don't have the whole genome sequencing for this one, but it's undergoing. And I just want to show you here that it's actually, it was collected northern, or more close to Brazil. So maybe my hypothesis is that this one is actually coming from the 2023 outbreak. probably. So maybe we are getting like all, you know, every time Brazil has an outbreak, we are getting some of the new variants that they are having there. So of course we will try to do some breeding. We started to do some breeding for this disease. So the first thing we did is, as many of you may know, the only good resistant out there is a translocated fragment from a wild relative, the 2NS fragment. And actually, some of the new isolates are breaking this resistance, but it's still effective for some of the isolates. So we have data for about 5,000 lines from the wheat breeding program. And we use a bioinformatic pipeline to predict presence or absence of 2NS. And we can see that only 14% of the lines had the fragment present, and almost 3,500 don't have it. So this tells two things. One, we need to be aggressive in introgressing the 2NS in our breeding program, you know, to be kind of safe on if blast starts to be more problematic, but also give us a lot of opportunity here to screen this germplasm and to see if some of these lines that doesn't have the 2NS shows resistance. Because right now there is not a lot of good resistance out there, at least in the primary pool, right? And so lastly, I want to show you this one. This is from 2023 season that we sent some materials to Capeco, to Paraguay, Mojancoli. And so he did some phenotyping for us. And so you can see here a few of the India varieties. And you can see here visual score 1 through 9 for the wheat blast severity. And what I put here is if they have or do not have the 2NS. And I want to show you that, of course, you know, most of the lines not having the 2NS are susceptible. But some of the lines here, like these two, they don't have the 2NS, but they have pretty good resistance in the field. So I think this is also giving us some opportunity to explore a little bit more this type of lines that we have in the program. So with this, I want to thank all the people involved, all the INEA technicians. So Fernando did the survey with me. It was a challenging day. I remember it was raining a lot. So we were doing survey together. And then Martina and Cecilia are the biotech people. Silvia Pereira, wheat pathologist, and also Diana run the phytopathology lab. Pawan from CIMIT, and Giovanna, Shenshen, Jim Stack, and Barbara from K-State. So thank you very much.